Hello, and thanks for tuning into this video. In this one I'll be explaining, going through the model that you're seeing on screen now, which is a geometry nodes setup on that allows you to create procedural microtubules. To sort of give you an idea of what the video will cover, so first we'll do a little bit of introduction on microtubules for people that are not familiar with them. Then I'll do a demo of what the model can do and what you can expect as features that are you can get if you download this as an asset. I will then briefly cover the geometry node setup, but it's not a full tutorial, so I will not go step by step over it, but I'll go over the general gist of it. And after that, I will show some sort of fun examples that you can do yourself, give you some inspiration on how you can use these this geometry node setup and modify it for your own purposes, and in the end I'll provide some instructions on how to download and use them after you've downloaded Blender, basically, so you don't need to know anything about Blender really to get started, although of course having some Blender knowledge in advance is always going to help you create scenes faster. But I hope that at least this setup can help people or result in people being able to include these rather nice looking 3D renders of a pretty accurate looking microtubule in their manuscripts, or just for fun to put in their offices or something like that. Okay, I assume most people here watching will probably be familiar with the microtubules, but I'll still just briefly go over them for people that are maybe coming more from the Blender side, and I give an extremely short introduction into what these are. So microtubules, they're protein filaments, so they're real things, although what you're looking at here, they look pretty big, but in real life they're extremely tiny. They're just, the diameter of this tube is only 25 nanometers, which is, well, extremely tiny, I suppose. But these filaments are found inside most of the cells in the human body, but in fact in many cells. Pretty much all cells with a nucleus, so anything except bacteria, as a simplification. So I took this diagram, that's quite a famous one in the field, that sort of outlines this. It's from the citation listed below. But these filaments that you can see here, they match this schematic illustration. They're made of these molecules that are called tubulin, and they're a dimer, so they're made of two parts that are always together. So you can see the two colors, the green and the white here. That's one dimer, so that's sort of the building block of this tube in biology. And this tube has a lot of interesting structural features. So typically it's made of 13 strands, which we call protofilaments, and they're set up in a almost helical structure, so not quite. So you can see there, it looks like a helix, but it doesn't quite. There's a discontinuity in the helix. That's called the microtubule seam, so that's also included in this in the model. This model has the seam, and then at the end of the microtubule, you'll typically, or in reality, often find that these strands at the end, they kind of flare outwards. So that's also been observed experimentally. So this model tries to replicate the structure or knowledge of what a microtubule looks like from experiments, and puts it in an easy way. But these filaments, I should also mention that it's also visible in this diagram that typically the molecules that get added onto the filament as it grows, there are a different kind that are in the lattice, so as they are in the bulk part of the filament. So as they get added, by chance they turn into this other type. You can see here, for example, that at the tip we mostly see green and white, but then as we go along, it's randomly turns to the pink type, and that's also trying to mimic sort of this process. But I think that's all to know really. They're the real things, but you wouldn't see them in real life because they're way too small. But it's very cool I think they're very nice looking filaments. That's why I chose to make them as a node setup. All right. The nice thing about geometry nodes is that it's a very easy to work with. So these, of course, there's a rather complex looking shapes, especially since they try to capture the flexibility of microtubules in real life to have they're quite plastic in their shape. So there can be the number of strands that make them up varies depending on the experimental conditions, but also the exact distance between the molecules and the shape of these ends. They depend a lot on the experimental conditions. So the model geometry nodes is perfect to capture that, and it's very easy to use because all of these that you're seeing here are actually they're just curves. And the curves are very easy to work with in Blender. They're just Bezier curves. So all you have to do is align a curve to your artistic desire, and then you can add a microtubule to fit to that shape. Meaning that also if you want to move one, you can just dynamically change it, and in real time you'll see how it updates to get it just right to how you want it to look. Alright, so that's a general introduction. I think now it's time to look at I'll go over the list of features that are included if you use the asset to play around with. Okay, so here I've loaded up a scene that I set up to show the features of the lattice, of the bulk part, where you see all these strands come together, and here from the top. So this is the same filament just curving around. So we can see the effect of the parameters on the right. So we have this whole long list of values. In fact, there's more when we get to the tip that you can control. So let's go over them and just see the effect of them, so you have an idea of what you might be able to change. Of course, if you don't like this kind of thing, you can just skip to the next section and play with it yourself. That may be more to your liking or how you like to explore things. But so the lattice spacing is straightforward. You change it, the dimers, the molecules making up the microtubule, they get spaced further apart. It still just tries to fit it along the length of the curve, so the curve itself does not become longer, at least not a huge amount, but the distance between them changes. We then also have the radius. This is also very straightforward, so it's just how far are the strands apart. If you want to be super accurate about your microtubule render, you would probably get an experimental structure imported into your Blender, maybe a part of a model of a whole helical rise or just a couple of dimers. So in the asset I've included a mesh for a tubulin dimer that's from a PDB file, and that's of a part of a lattice, so you could load that in. There's tutorials on YouTube from Brady which I'll link right now on the screen, that you can use to import such a PDB file into Blender and work with it there. But you could use that as a reference to tune exactly how big the radius should be, how the lattice spacing should be. But you could also leave it up a little bit to your artistic choice, so to say. Then, and obviously we have the protofilament count, so you could change how many strands should make up this filament. Typically in cells you would find 13, at least in human cells. But, let's say 14, 15, 12, they're all fair game, according to biology at least. So you can set them here as you like, probably adjust the radius a little bit as you change it, but that's super easy to do. Then we have a lattice twist. So it also does what it says on the tin. You set it, the lattice twists along, and of course the end goes crazy because it has to incorporate the whole length twist. But it just twists the dimers, but maintains their orientation along the spline shape, basically. Now, if we're talking biology, then the actual twist that a real microtubule would have is extremely tiny, maybe something like that, or that. So really very hard to actually see. But they are twisted, some depending on the kind you're looking at. But you can, for dramatic effect, right, if you want to show something about, you do an experiment involving twist or measuring twist, you could show for dramatic effect, demonstrating lattice twist. Here, set it back to 0. Then we have an important feature of a microtubule, which is the rise. So a microtubule is not just a tube, it's kind of like a helix. So if you were to set the rise to 0, now it's just a tube with nothing on it. If we set the rise to 1, so this is a scaled rise, so it is scaled to the number of protofilaments and the lattice spacing. So if we set it to 1, it's going to rise exactly 1 lattice spacing after a full rotation. So as you can see now, it rises exactly 1, and that makes it a perfect helix. This might be the case for a 12 protofilament microtubule, but the most common kind of microtubule is actually not a perfect helix, as I mentioned before, because its rise is about 1.5, meaning that when it comes around, it doesn't go up 1 monomer or 2 monomers, but it goes up 3 monomers, if you will. And that means that the different types, indicated by the different colors, they don't match up. And that creates the structure along the back here, which is called the microtubule seam, so that you can generate that microtubule seam simply by having this rise set to 1.5, which is appropriate, at least for the most common kind of microtubule. Of course, you can set it to something crazy, make a monster tubule. Not sure why you do that, but yeah, if you go very large values, it starts to get a little bit unstable. But you could set it to 2, I don't know of any microtubule structure that has a rise of 2, but it's probably possible, given how much they like, how many things are possible with microtubules. Let's see, is there anything else that we have to look at here? No, so that's the parameters that make up the lattice. But of course, one thing I forgot is that you can set the model that you put here is up to you, right? It doesn't have to be this mesh. Maybe you like it a little bit more low resolution or more abstract. You'd like some kind of sphere, or maybe a deliberately blocky shape. That'd be quite nice, actually. So we could set something here. I have a small cone that I use. This is also included in the assets because it's quite a useful shape to check if all the orientations are correct, because you want this stub, this little pokey bit on the cone, to be facing outwards. And with this shape you can really easily check, because a tubulin dimer with a realistic mesh like I just had before, it can be quite hard to tell if it's oriented right, and you can miss it quite easily. So you probably, if you're making a render that's quite important, where you're really, you're putting a lot of effort into it, you probably want to take some time just to check it with this shape to see if everything is proper. In general, all the dimers are oriented correctly in my experience, but every once in a while there's one dimer that just happens to be, I guess, on some kind of rounding, a point where the curve rounding is a bit off, and then it faces the wrong way, and it'd be kind of unfortunate to have that, in a pretty render where you'll notice it later on that, oh, this one thing is not oriented right. Anyway, but you can see you could put any shape here. You could put a little sphere here, or a cartoon sphere, to make it a very minimalist kind of simplified drawing or render, but then still have the full complexity of an accurate 3D representation of the microtubule, which can be quite hard, at least for me. I find it hard to make that in a 2D sketch program, because thinking about this curvature in 3D is quite a challenge. Alright, so that's the lattice flexibility. So I think it has everything that you would need for at least a canonical, let's say properly behaved, intact microtubule lattice. So now I think it's time to have a look at the tip. Okay, let's go have a look at what's possible with regarding tip shape in the geometry nodes microtubule. So let's go to the basic shading mode, so my computer doesn't catch fire. So here we have two microtubules without a tip. So this is not particularly realistic. You probably wouldn't see anything like this in a cell, but in the model you can see this value here, which says flared end, and that's just a Boolean toggle for the end shape. So we set it to 1. Now we have some flared end, and 0 turns it off. So that's quite simple. Same here, we could just go there, yoink, get a flared end. Of course, driving this to get a shape just how we like it, we have a lot of parameters to play with. So first up actually is the end type. Now this is, if you notice, it changes a little bit the mesh, and actually what it does, it flips the dimers sort of backwards along the curve, and what that does is it essentially makes it a minus end. So assuming you set up your dimer mesh correctly, now this would be a minus end of a microtubule. So that's the other side, where obviously the orientation of these molecules is flipped. So let's grab the cone as a demo again. You can see quite clearly, changing the end type simply reverses the model along the curve, making it a minus end. So that's a straightforward feature. Let's set this back to a dimer mesh. Then we have two parameters related. One is the mean length, which does what it says in the tin, but you can drive the mean length. Okay. But also the mean length variability. So if we set this to 0, now let's set everything else actually also to 0 just to get started with. With the mean length variability to 0, you can see they're all the same length. Again, might be interesting for some abstract representations, but in real life or in cells, they would not all have the same length because they're not somehow feeling each other's effect. So there's some variability in the length. So that's what you can do with this parameter. And then you can tune it again to your liking. So now we can see some are long and some are short. So you can play around with those two values to get the ends to look as random as you would like. So I'm going to tune this maybe to 0.3, maybe even 0.2, and make them a bit longer, something like that. Quite like that. Then we have a parameter called raggedness, and to show you why that's important, let me move this one out of the way, is let's say we set this to 0 and make them long. Now they all curve at the same point, and that's also not very realistic. There's nothing magically inducing a curvature at this point. So with raggedness, it randomly offsets the tips of the microtubule, and therefore also the point where they bend. So now you can see this one's bending here, this one's bending there. So now it looks a little bit more realistic. You would not expect them to all curve at the same point. So again, you can season to taste, as we'd say. to how much you like it, and balance these parameters. Then we also have the fluctuations, which is just very simple. It takes these endpoints and it just adds a random deflection to them. So as an example here, well, probably don't want it that extreme, but something like that, still a bit extreme. You can see now it looks a little more, organic. They would not all be perfectly curving out. There would be thermal fluctuations and random bending of these protofilaments. Yeah, so that's what that does. Fluctuations. Then we also have, again, let me set this to 0, parameter for rotation, which just twists them, the end, a little bit around. So this has also been observed experimentally, that this is a thing. They tend to twist one way. So you can add that with this end rotation parameter. Yeah. Again, just to give you that flexibility of how you want the tip shape to look. Depending on your angle, you may not notice it, but it's there as an option. And then finally we have these 5 parameters: flare tip radius, flare tip length, flare tip angle, flare base length, flare base angle. And these define the shape of these protofilaments, of these flaring tips. So the radius is relatively self-explanatory. It's basically saying how far is this tip out from the lattice. So we change it. You can see now they get closer, and that's all it does. That's just the position moving normal to this along the curve. So normal. Let's set this back to 5. The tip, and then the other 2 parameters, tip and base, refer to this point. This is tip. Maybe blue's not the best choice for color. Can make that red. Yeah. Tip and base. So these 2 points, and each point are control points for a Bezier curve. So we have, let's say, handles like that. And we can change the length of these handles and the angle of these handles. Right? And with those values, this distance, that length, and those 2 angles, allows us to cover quite a large range of tip shapes. So for example, if we make these 2 handles smaller, shorter, we'd expect this curve to be less domed, let's say. So let's make it shorter. This one also shorter. And there you go. Now you can see it's a much flatter curvature. Let me get this back in projection. Okay. But we can also change this, the effect of the tip angle. Oh, I'm not sure how pronounced it's going to be at this length. Okay, let's make it a bit longer. Tip length, tip angle. Yeah, so you can see the effect there, changing shape. Or we can change the base angle. See, now they're a bit closer together at the tip, or more outward. This doesn't look right, but that's okay. Or we can try to get them to curve more like that, more outwards. So that we probably want a long tip length and a large tip angle. So there you go. Now they're curving around. Now I find them to be getting too far apart here, so I changed the space angle a bit back. Yeah, that looks better. I mean, again, I wouldn't say this looks accurate, but it just shows you what you can do with those values. Yeah. So that's all these things you can do with a tip. So a lot to play with, a lot to tune to how you like it or what you think is most accurate, depending on what you need. I think some artistic leeway should probably be, given, especially since we don't fully know. Well, there's a lot of variation out there in the biology, it seems. So then, perhaps whatever you draw is true in some instance, somewhere, at some point in time. Yeah. So these you would probably set at the start of your you're working with, of your sample, unless you want to really individually tune the shape. But how I would use it is just you find a shape that you think, well, this is how a tip should look like, and then you change it with these parameters. You change it a little bit, try to get the variation in there between microtubules. Because if I duplicate this object, we come to an important next parameter, and that's the randomizer seed. Because a lot of these parameters have a randomness to this. The raggedness is based on a random number. The length is based on a random number. If you have a end variability set, the fluctuations are set on a random number. So if we change the seed number value for the random number generator, we get a different tip. So we can easily get unique tip shapes, but which are still in the same family because they still have the same sort of underlying curve shape, but they all look different. So I could now have a family of 3 microtubules here that all look like they're grown in the same condition because they have the same kind of curvature of the tip, and they all have the kind of a similar mean length and a similar raggedness. Okay, this has 0 raggedness. But you can easily generate multiple of them just by changing the randomizer seed. So you see, you can generate many tips that, if you set the model up how you like it, are probably going to be something you're satisfied with. And if you get if you end up with a tip that you don't like, well, you just go through a couple more randomized seed numbers until you get something that you think, this looks nice. I like these 4 together, but this one crazy one here, just keep it at that. Of course, all these parameters can be animated if you so desire. I must mention that while you can animate this model, it's not necessarily set up to facilitate animation. So one thing I did add that you may notice, let's grab this guy. But if you extend it, it actually does look like new bits. Okay, maybe I should move it towards in the orientation that I'm dragging it. When you move it, it does look like new dimers get added to the microtubule, which is nice. It's not perfect. It's a little bit twitchy every once in a while. When you try to animate this, it's clear. You see that. I think that's because it has to sample this backbone at some finite precision, and then it slightly changes how the curves act depending on the movement. But in a pinch, you could animate it like this. It looks all right. It looks acceptable. Not perfect. Not like studio quality, but good enough. And also, as it extends, there's no variation in the lattice, in the tip shape, so you probably have to do that manually. And it's not very easy to smoothly change parameters manually. It would be quite all or nothing changes. You could try to fix that by changing the random number generators to noise texture, which are sort of smooth in space. But in general, I would say if you want to use this for an animation and you really want it to be high quality, you may need to do some more tweaking with the random number generator to allow smooth changes, because having the tip always look the same as it grows, that just doesn't look right. Alright, then I think we've covered everything in this list except for these two values, the hydrolysis onset and the hydrolysis length. Now these rely on you having set these two meshes to some different materials. If you set them to the same material or same object, you won't see you won't see anything here. But let me turn that off. Turn off the interface. And the hydrolysis refers back to this feature that I mentioned in the introduction, that new molecules that get added to the growing end of the microtubule, they are of different kind. So that's in the back, and this randomly turns into this type over time. So this feature and this drop-off is then, well, I've modeled it here as an exponential drop-off. But it's set by these two parameters. The first is the length, so that's just how quickly does this pink type, your GTP type, fall off over space. So if you set it to a very small value, 0.2, now it falls off quite quickly. Set it to a very large value, 2, now it takes a very long time to fall off. There. As you can see, there's a little bit of blue starting to appear here, but it's still quite far away. Set it back to 0.4. That looks quite nice. But what you may notice here is that at the tip, there's some GDP, or blue molecules, and that's not very realistic. Or maybe you'd even want the change of lattice type to start somewhere here. So there's another parameter for the onset, and that's just, let's say we set the length scale to 0. Now you can really see the effect. It's just a linear offset, no randomness to it. Where does this exponential fall off start? So you can use this to make sure, for example, that your tip will never have any blue molecules in it, just by setting this offset value. Keep in mind, setting it to 0 will leave your tip molecules, all your tip molecules, to the wrong type. So you need to set it to minus 0.1 to really ensure it's all gone. But that's the smallest value I've allowed in the range anyway. But then the length scale, so for example, I might say I want the lattice to be GTP only in this area and then fall off a little bit over the length, and that's what you have. So you have a fixed area here, exponential fall off here, and then mostly GDP now on this end. And of course, I mean, if you, right now, in this example, I just have the model, the same model with a different texture, different shading on it. But you could, in principle, have something a completely different model here. So you could have, let's undo that and set this to our trusty cone shape. And now that's quite different. Or you could even have it, like I showed before, have it not there at all. I mean, I'm not sure why you'd want to do that, but you could. Or set it to something cool, like a transparent material. Would also be quite neat to do that. Yeah, and of course you can hook up into that, and I'll show you a cool demo later on about using this hydrolysis to selectively only bind molecules to the GTP part, for example. Again, I should mention that this is not a very animation-friendly feature because I switch this back on. It's sort of, as you move it, it recalculates the random number. So it just completely changes every time a new dimer gets added. So there's no history, there's no memory of what turned to what before. So for animation, it's not going to look right. Again, in a pinch it may look right, but it's more really for static images, then it looks okay. Alright, let's go have a little look at the node setup behind this, the Microtubule. Now keep in mind, this is not a full tutorial, so I'll just kind of do the general walkthrough, and then I suggest, if you're particularly interested, you just download the model and look at the details, or try to reproduce it based on my description. So we have a basic Microtubule setup here with some a nice looking tip. Let's go to the nodes. We grab our geometry nodes panel, and here I've set up what we've just seen, but also another one where I've disabled all the nodes so we can see step by step what happened. So let's make this a bit bigger, close that, and here we have the Microtubule. So it's a big, obviously, pretty big model. I've set my as a detail, I've set my line interpolation to linear. I find that a little bit easier to follow than the curves, but that's subjective. But if it looks a little bit weird or different to you when you look at it on your computer, it's probably because your line interpolation looks a little bit more like that, more curvy. Anyway, so let's start by our setup over here. We have nothing, just the base curve. That's how you would draw it. So how do we get to a Microtubule? So we just follow the curve. It's laid out such as kind of left to right is how the model flows, or how the information flows. So we start off with this is our spline, our original backbone, and we start off by just resampling it so we get a higher resolution. So this may be, depending on how you set this up, just 2 control points, just the ends or more, but it could be just 2. And to get a high resolution information for these points to put the dimers on, we need to resample it. So we just sample it by length. Here I do the lattice spacing, but it's not absolutely critical at this point in time. It just has to be high enough. And then we capture a couple of attributes for later use. So we capture the position, the normal, and the tangent of the original curve that you've drawn. And then we do a trick that is based on a video which I'll probably link in a card right now, where we copy the curve n amount of times. So we just create a simple a mesh line, basically, at the same position, and we just make the number of protofilaments that you've set copies, and then we copy the original curve that n amount of times. Of course, it's all still at the same position, so now we have n curves. Let's say 13 from now on. That's probably the value you'll be using most of the time with this model. 13 protofilaments. So we have 13 lines all overlapping in the same point. Then we capture their index, so we can use this later on. So we can selectively play around with this value. For example, if we only want to do something with the first protofilament, or number 3, well, we can use this captured value later on. And in fact, we'll need it later on as well. But then we realize the copies, because right now, after this step, they're just instances, but we want them to be real things, so we can manipulate them and not just move them around, but do more to them. That's all fine because they're just curves. I mean, 13 curves are not that computationally demanding. And then we make them a Bezier type. So I think in one of these steps, it may reset it to a polyline. Blender seems to do that. Well, we just force it to be a Bezier type here. Bezier type means it can have these nice curves, basically. A polyline is literally just straight lines between the backbone points, and that's not really what we want here. So that's all good. Now we just have many curves at the same point. And then the next block here is all about making these flared ends, putting them on there. That's the purple block. So there's a lot going on here, but let's just take it step by step. First we have these selectors that are very important because we want to have a relatively basic control the shapes of these protofilaments with just 2 control points. So what we do is we take the original spline here and we take the end point and the 1 point just before the end point, so the final 2 points of the curve, and we just make these shapes with those 2 points. So we have last end point, last and second to last end point, and the second to last end point separately. So these are the 3, this is the last final point, the 1 point before the end, and then the 2 combined. These are selectors that we use to move things around. So first we set the position of the tip, so that's the final point. We move that some distance away. Let's enable that. So you can see it here. We just move them out some distance. Of course, this doesn't look like anything, but as you notice that it doesn't really matter when we're looking at splines because it just connects them. But this is a point, and this is a point, and anything in between, there's no points in between. So this is it's a low density of points in between these. Doesn't look like anything, but now we also, then this node here handles the raggedness, which actually offsets the final 2 points, that's this point and this point, and moves it in Z, or along the tangent of the backbone, some random amount. So that's just a bit of a realism thing. So there you can see it just moves these a random distance. Still doesn't look like much, maybe a very abstract tree. But now we start to use these 2 they move the handles of the endpoints. So it's all about actually making use of that Bezier curve type. Now, so we here we set the handle basically of the tip. So that's this point. Now we're going to move the handle around, and you can see now, you can really see the Bezier type coming now. You can see a nice curve. It still doesn't look right, but it's starting to look like something. And of course, this is all controlled using some vectors related to the backbone that you originally drawn. Here they're also already rotated. We'll get to that in a bit. But now we're almost there, and then we also, for this point at the base, let's quickly disable that. For this point, we also need to now set the handle, because right now it's just doing nothing. So we get this kind of weird curve, and we want to be able to control not only this curve handle, but also one that sits here. So let's enable that. There we go. Now that's looking more like a rather extreme, but Microtubule tip to begin. Okay. And now, of course, if we change these values, so we can now change these using the parameter panel here. So we could change the radius, the tip angle, which is related to this Bezier handle angle, but also its length. So as you can see, it changes we just changed the handle of the Bezier curve for these 2 points. So that's fine. And then we have a final thing here, which just changes the length of the curve. So a real Microtubule doesn't have all these flare tips at the same length. It'd be quite random. So we just randomly trim them to a certain length. So here they look quite even, but we can set the end length variability higher, and now you can see that the end length is quite different between the different curves. But now, if we were to now put points onto this without resampling the curve again, we'd have a diameter here and a lot along the backbone here, many here, and then one more here, but nothing in between, because this whole bit is just 2 points, whereas this whole bit is many points. So we resample the curve to fix this issue. So now again we will have an evenly distributed number of points along this curve. So now the density of sort of points that make up this line is even again, uniform. So okay, that's how we make the tip. But as you can see, they're all at the same point in the middle. And obviously Microtubule is a hollow cylinder, not some kind of crazy thin flower with flares coming out there. So that's what the purple block does. It consists of just a single sort of geometry manipulation step, which is a set position node. But there's a lot going on here. But basically, it's taking the curve normal of your backbone and then rotating it around the tangent. So you get a vector out here. Let's see, can we draw this? You get the normal, and then you rotate it around the normal like that, and that's how we can get the hollow tube effect. And then we also add a lattice twist, which sort of means that whereas the next part of filament might be here, and normally in the next step, one dimer up, it should be slightly more rotated this way, and the next step slightly more that way, so we get a twisted lattice. Anyway, I suggest you have a look at the node setup if you're particularly interested about how that's done. But let's enable it now. There we go. Now that's looking like this already, although I've changed some of the parameters here about the tip shape. But now we can recognize the Microtubule. We have these splines nicely offset, and yeah, that's how you probably would imagine in your head how you would draw a Microtubule as a set of splines. All right, and then the next step is very simple. We capture another thing, the spline parameter, which we'll use for the hydrolysis. And then it's just a matter of adding instances, instantiating the dimer model, the tubulin mesh that you can set over here. So you can have whatever model you like, of course. Adding that there with an instance on points. Now, critically, what you may notice is I do not add a curve to points after this point, and that's very important, because if you were to do that, we'd run into a problem that I will show you with this example. So here we have a simple curve, and we have points along the backbone. So let's grab this demo node, and we do the same trick. We let me get a normal view. If we were to take the normal and rotate it around the tangent of this curve, and then offset it, all these control points, offset them along those vectors, then we'd get something like, oops, like this. And that's great, because you can see along the curve here, the distance is pretty big. It gets bigger, as you would sort of intuitively expect, and along here it gets smaller, and that's good. And critically, the distance here between those points and these points is matched, because it's all offset from the same origin point, and then it's offset and offset. So in the parts where there's no curvature, they're nicely matched. But the important thing to realize is that this curve and this curve has a different length. So if we were to then resample the curve later, so that would be either a resample curve node or a curve to points node, now we'd be computing this based on length or count, but it would place it uniformly along the curve length. So it would figure out the distance between points and then distribute that along the curve. But that immediately presents a problem, because we do not want a uniform density of points on the curve. Because here you see the problem. All these curves now have the same distance between them, all these points, here, here in the middle of the curve, and here at the base. And I mean, that looks fine if you just look at one curve by itself. And also, again, if you look at the bottom, it looks nice, uniformly spaced. But you see the problem. These points, especially in the part where it's straight and you would not expect while you wouldn't want an effect of the curvature all the way over here on the spacing here, now these points are closer together than the ones up there. You see? And that's just fundamentally wrong if you want a microtubule model, because you wouldn't expect. It's very important that these dimers or these molecules that you set, they sit at the same relative position to each other. So we can't have suddenly that the dimers here are closer together than there. So originally I didn't realize this in my original, at some point during the model design, and then I got into trouble and was confused. Why is my is the microtubule seam an important feature of the microtubule? Why is it not lining up far away from the base of the microtubule? And that was because of this problem. I had a curve to points node, and it was resampling this node setup. And because these curves are different lengths, you run into fundamental issues, and so you want to avoid that at all costs, if accuracy is important. So let's go back here. Let's grab this. So actually, originally I had this purple part come before the red part, but then the problem was I had to call this red part does this offset from the backbone. So after this, we do not want a resample curve, but after setting up this tip shape, we do need a resample curve. Otherwise, there'd be no protofilaments here. So anyway, there was a whole thing to make sure this one goes before this one. But anyway, then we simply add the dimer. So here's the GDP and the GTP, and there we go. So let's see, things are going on here. The rotation is also an interesting point. So this little red box here handles the rotation, orientation of these dimers. And they're separate parts. So we treat the lattice, so that's the normal part, and the tip, we use a different vector basically for handling how these dimers are oriented, because the spline itself, so the things that you would see here, these are not reliable enough to use. Their normals are not reliably aligned along this curve. Well, not for any arbitrary orientation at any rate. So for the lattice, we just take the vector that we had from the backbone. So that would be, let me draw on this. We take this vector that we rotated around the tangent. We basically take that vector and we orient it face outwards along this. So that's pretty straightforward and very stable. Although every once in a while, one of the dimers flips, but yeah, it's quite rare. And you just need to twist it slightly differently, then it's fixed again. But for the tip, it's a little bit more difficult. So for the tip, basically, what I do is we have so the base of the microtubule has, we take the vector outward that we use to in this step. I'm drawing here the vector that handles this change, and then we offset it to sort of a point that's right in between here. So it's in between the base and the tip. We say, okay, we generate a point there, and then we take the vector that points from all these dimers that points that way, and then we take the component of that vector that's normal to the tangent. So maybe let's twist this a bit. We have a midpoint here, and maybe the vector for this dimer points that way, but the normal of the curve points that way, meaning that this vector and that vector, they're not normal to each other. But we take the projection, the normal projection component of this this vector, this one, compared to this normal, and then we rotate the faces first along this way and then the z direction along that way. That's what we do here. Here we generate a normal vector relative to the tangent, and then we orient the x-axis, x face of the dimer. Oops. Oh, I can't disable this in this mode. So we orient all the faces, x faces of the tubulin model towards facing the normal component of the tangent, and then we put the x component, and that does it. And that seems pretty stable, so I haven't had any issues with that. It's the most stable solution I've come up with that kind of seems to work with every tip shape and how crazy curvature that you put on your backbone. Fine. Yeah, that most of it's there, but then the final thing is the hydrolysis model. So for that, I reckon we need to go to rendered view, because otherwise it's not going to show, and I'm not entirely sure. Oh, I need some other model. Let's grab maybe Can't forgive this mess of a node mesh setup. Okay, maybe it's not very clear still, but it'll do. Then we where's my hydrolysis onset? So let's set this all to 0, 0, and 0. So now everything is made of the GDP model. The hydrolysis is very simple. We just take the spline parameter, which is spline factor to be precise. So that's a value that runs from 0 from the base of the backbone to 1 for the tip of your curve. We capture that before we add the dimer, so we have this that's for each curve independently, for each tip shape. So 1 for this curve, 1 is here, and for this curve 1 is on this end. It doesn't matter that the original curve was longer. And then we essentially just do an exponential decay function based, some factor based on exponential decay of this spline parameter. So we have an offset, hydrolysis offset, that's sort of just a start factor, and then the length scale, which is sort of the power in your exponent that dictates how quickly these fall off. And then, of course, this exponential curve just gives a probability that any dimer is going to be GDP or GTP, and then we just check with a random value node whether a particular dimer is going to be, well, which type it's going to be. So that very simply, and then out of this comes a Boolean selector, and then we just simply plug that into the instance GTP, and then the inverse of that, into the GDP. So we're always certain that we don't have any overlapping structures, and then we combine we add a joint geometry, so we separate these two. You could theoretically do this with some kind of set material thing if you don't care about having them be different meshes, because I probably wouldn't bother taking a different mesh for GTP since the difference is so small. You probably only want to change the color for visualization purposes. But the problem I had with the set material node was that it acts on a face level rather than an instance level, so it was more efficient to do these separate instances for my case. So that's the whole thing, and then this little area here is just for you to play around with if you have a cool idea about some custom things, you want to put molecules on there. I have a couple, I think later on, there'll be in the video, there'll be more, some examples of that, what you can put in this box to make, add some things to the microtube that interest. But that's the whole thing. So it's, in summary, it's resample the backbone, add copies, add the tip shape to it, and then offset the tip shape from the backbone to create a hollow tube, and then add the dimers, and then these two auxiliary groups just for rotation and hydrolysis. there's a lot of things, little problems that went in and solutions that came along that might be interesting for you to look at. But at the end of the day, that's the general gist of it. Alright, to use the asset, you first have to download the file listed on the link in the description, and after that you simply go to open a new file like this, the standard cube, file, edit preferences. In your preferences you'll navigate to file paths, and then down here you have asset libraries. Click the little plus icon and then select your path. So just navigate to wherever you have saved this download. Suggest some kind of folder where you can find it again and not just your bulk downloads. And then you give it a name that you can remember, something simple, maybe Microtubule, that'd be fine. And then you can close that, and now from here on out, this Blender installation will have this asset loaded. To then use the asset library, you drag another panel, another area I should say, this, and you select the asset browser. Now we still don't see anything, because right now it's just loading, trying to find assets in the current file. So we take the drop down, and here should be whatever you have listed as name, what name you just entered for your asset, and I name and you carry it, click that, and here you see the models. And since running Blender 3.1, we'll take the proper one, not the old one. And now it's just as simple as you just drag it into your scene and let go, and there we go. We have a Microtubule. Fantastic. And this will be like this for every new file that you make, and you can just use it as such. I will say, once you have one in your scene, you should make more by duplicating the one you already have rather than dragging one in. So just Shift D to make another one, and then you can edit that however you like. Oops. Yep. And then because if you drag another one in, it does work, but it means now it has extra copies of these timers, and so it's like a separate independent instance of the Microtubule, and you want them to be sort of linked, all share the same underlying objects. And that's it. So it's super easy. Just open your asset library and just drag and drop, and you have a Microtubule in your scene. It's very nice. Of course, you can combine many of these different asset files into your system. So if you've got another fun tutorial, or you made a really interesting node setup, you can make your own asset and have all these different assets ready to use whenever you want. So maybe one for a quick one for a membrane, another one for actin filament, etc. So you can quickly build up a scene with your own asset library. Do a little demo for binding Microtubule associated proteins to a Microtubule. So I've loaded up the standard model, disabled the tip for clarity. As you can see here, I've flared to 0, and I've set up a little bit of a GDP hydrolysis over here. But let's say we want to bind maps along this lattice. Let's also set the twist to 0. Well, I've loaded up one map, EP1 model, and Suzanne, which are listed down here. If I can, I cannot click that. All right. So this Suzanne and this EP1 model. Now, of course, I can manually go position them along the lattice, but we're trying to do everything procedurally to make our lives easier. To do that, it's actually very simple. So we can use collections. So here are collections, our Blender feature. I've placed the things I want onto the Microtubule into a separate collection. And I must mention that a collection has its origin, although you can change it, but it's a bit opaque, and how it's done is not very visible to the interface. Has its origin at 0, 0, 0. It's a bit involved to change that, I think. But let's just get started by first just getting these onto the lattice, and then we'll worry about the exact positioning later. But we want these on the lattice. So we'll go to our geometry nodes, select our microtubule, and we're going to have to do some magic in the maps and more area. Well, this will be here by default, a collection info node, so let's select our maps, and that just loads in the geometry of whatever objects or light sources even are in maps. So then we want to instance these objects in the collection onto the location of these dimers and then add them to the structure. Okay, well, that's very simple. So we go instance on points. There we go. Doing this, we'll just add some points. Let's go back to this view. And then we chuck in our geometry and make sure it's set to original and not relative. And we also want the rotation of the dimers plugged into that. Okay, so this already gives a very good fit, but I want to mention that these might be at a random location. And then you can see, okay, it creates a very psychedelic looking microtubule, but this is not obviously how you'd want your molecules to bind. And the reason why they're offset is because their location is relative to the center of the dimer, or essentially the point at which the dimer is instanced, and then rotated with the same rotation that is given to this dimer object. So clearly these are offset some way from the origin. As I mentioned before, these collections, they have their origin is at 0, 0, 0. So this Suzanne head is offset from 0, meaning that if we instance it, if the center of the dimer is here, it would be put all the way over here. As we can see, it's pointed out in the EB1 model. It's even further out from the origin, and hence it's put out even further from the center point, which is the center of each of these 2 billion dimer molecules. So the best way to do this, I find, is to just take your GDP molecule, so you need these in your scene to have these selected for your mesh here, so they're going to be in your scene anyway. Just put them at 0, like that. You know, they might be at a random point in your scene. It doesn't matter where they are. Put them to 0, and then you can start. Just simply center your camera maybe on the dimer and position your molecule how you think it should be binding to the microtubule. So let's say the Suzanne molecule only binds to this beta tubulin, I guess the beta is the top one, and it binds face outward, and maybe we'll make it look slightly down like that. That's how it should bind. Okay, let's see what that did. So if you scale the object, make sure you apply this scale afterwards, so it's set to 1 here. Otherwise it may start doing some weird things. But now if we look, you can see that perfectly this location relative to the dimer is replicated precisely all along the lattice. And of course it's maintained as we twist the lattice or change the lattice spacing. It's all maintained. And as we bend the thing, it will also be maintained, as you can see. So that's nice. And we might do the same thing for our EB1. So we center our camera with numpad dot, full stop. Position our EB1. The rotation of EB1 here is probably completely wrong, but let's just say it binds exactly at sort of this interface on the side a little bit. And then we can see indeed that if I change this down back to realistic value, now we have our molecule binding exactly at this interface. So that's all good. You can see it's all replicated faithfully. If we go quick rendered view, yeah, looks nice. A bit of a clashing color scheme, but certainly acceptable. But maybe you don't want them to bind everywhere. So let's say maybe we want only 10% decorated to make it a little bit more realistic. So the way we could do that is by using this selection port and working with that. So we could add a random value just between 0 and 1, and then we say, okay, is it a math node? You then say, well, let's check if this value is less than 0.25, so that's about 1 in 4. This will be true for. Plug it into selection, and there we go. Now only 1 in 4 molecule dimers will have something on it on paper. Yeah, I guess that's about right. Or we could change it to half. Now half of them supposedly have a timer bound to it. Maybe 80%. Look at that. Very busy, crowded lattice right now. But with that you can sort of, oh, that's a very non-smooth slider. But you can get them to bind only in some areas. You could even consider hooking it up to something like a distance thing, so that might be nice as well. Let's show that. Undo that, and we'll add an empty sphere. Oh, not to the collection. As you can see, if I add anything you add to this collection will be added on this position. So that means empties and lights, they all get added to it. So if you add the wrong object into this collection as your primary one, which you can change by, I think, clicking this icon, it'll get added to this. So if you have a massively complicated object and you accidentally chuck it into this selection, your computer will spontaneously catch fire. Well, not really, but you'll have a bad time unless you saved your file. Okay, so we have our empty, which we'll use as a control object. Let's scale it up a bit and have it there. Okay, so now what we want to do is only have these molecules bind to the lattice where sort of within this sort of empty sphere. So let's get back here. We need something about this sphere, so the easiest way to do that, take the empty, we'll call it controller, pin this one so it doesn't move when we select the controller, drag it in here. Okay. So we want to do something based on distance, so we got a vector math distance, and here we want the location of the dimer, so we grab object info location. Oh no, what is it? Position, my bad, it's the position field. So this now has the distance. When we connect it here, it would give us the distance between any of these points, which is the points where the dimers are instance on, and this orb. And now we want some condition that math, if it's smaller than, less than our scale, so that's the size of this sphere, then we want it to be true. So that's what we have right now. If it's within this sphere, it's true. Plug it into selection and set this to relative. It's very important. And now, as you can see, where we have our sphere is where we have our molecules. So that's very nice and intuitive. Again, you could do the same thing. Let's combine it a bit. Chuck in an and. Oh, that's not in here. Right. Boolean math. And selection. Good. And then again we do random value. Random value. Smaller than. Greater than also works. There. Oh no, never mind. It doesn't. Math. less than, don't clamp it. Well, about half, so now you can see about if any object in this sphere within our controller object, half of them randomly get populated with molecules. We could change it with the seed, get a different random number, a random number seed. So that's quite nice. So you can, make a nice art scene where your molecule of interest that maybe you're doing research on or something like that is bound to the microtubule at more realistic density. Because if it's everywhere, then, well, you have to be a very talented, a very good crystallization protocol, I guess, to get that to work. But this might be more realistic, maybe 0.2. In fact, you can tune it to your liking and then render it. Have something nice to go with it. Now, lastly, let's retire. Maybe, okay. What if we look at our lattice here, we see that we have GTP. Maybe we only want our molecule to bind to the GTP dimers, but not to the GDP lattice. That could be something that's biologically relevant to your system. So let's go back here, back to the state where we have everything. Because what we have here, for convenience, we have the GTP selection handle actually. So we can use this to say, well, let's just do GTP. As you can see now, it's only binding to the GTP molecules, dimers, and none of the GDP. And as you change your hydrolysis length, for example, it of course automatically adjusts because it's all procedural. So there you have that. Okay, very nice. And as before, you could do the same trick. So let's say you only want 10% of these molecules or a quarter of these. You simply add another Boolean math node with an AND. Let's remove that. Random value. Math add, whoops, less than 0.2, and now only, or for example, to 2.5, and now only half of them of the GTP things have dimers have a map bound, but none of the GDP molecules actually have one, because that's already set in our first condition here. And of course you could sort of, if you want to do only GTP, just add a Boolean math and a not node. Why is this now binding nowhere? No, it's only binding here. Interesting. Let's change the seed a bit. Okay. Yeah, so now you can see we're only binding just in some random points to the GDP, but not the GTP part of the lattice. Yeah, so there's a lot of things you can do with this, and I'm sure you can come up with very creative solutions here for how you want to arrange it. But I hope this gives you a bit of an idea of how you could combine sort of your more manual positioning for getting a nice render and the procedural tools for getting a certain density of molecules in the lattice type that you want, and many more. I think one more thing we could do, just for the demo's sake, is we could see if we can only get it on one protofilament. So we should have, it's not there by default, but we have it here. So we could drag this in. I think that should work. Add a reroute. Okay. Let's just get rid of these existing ones. Add math. Oof. Less than, so less than 2. This is the protofilament index, I think, running from 0 to as many as you have. Yeah, as you can see now, we only have protofilament 1 and 2 decorated with our dimer. Can we set it to equal to? I am sure it's there somewhere. Compare? No. Okay, well, we'd have to probably combine two of these to get any protofilament to show up, but just like that, only on one protofilament, and of course as you twist the lattice, that is retained because that's it runs along the same spline. Let's twist it the other way. There we go. Yeah. You can see now it runs along this protofilament. Only there. There we go. So lots of fun to play with, and try to see what you come up with. Alright, let's do a little demo, this time just showing that the Microtubule each dimer can be adjusted individually. So for that, we'll grab our asset browser as always and get a Microtubule to work with. Alright, let's put that a little bit closer to 0. Let's put it in a little bit more interesting orientation. I think that'll do. Good. Close this again. Then we want to go work in our geometry node setup, but first we need a control object for this demo. So let's add something that we can move around and then move deflect the dimers as if somehow something damaged the Microtubule in that location. So we'll add a empty, doesn't matter what it is, it could be any object in fact, but I like the empty sphere. Let's give it a name so we can recognize it. The Holy Hand Grenade. There we go. That should be catchy enough for us to remember. And now we can go into our geometry node setup. Now, in the Microtubule geometry nodes, let's pin it. There's this little area that I left open for easy custom adding of things to it. So we can use that to work with. So first we need to somehow get information about our Holy Hand Grenade into the setup. I could either add an object info thing or just straight up easier to just drag it in here. So there we go. We have our information here. Okay. And now what we're going to want to do is somehow, based on the distance of this object empty to this dimer, we'll move them to a random location basically, and we'll make it depend on the scale of the hand grenade. That would be nice. So first we're going to have to do something about position, and so this is where it all comes together. So after that we'll add a set position node. Nothing happens of course because, well, we're not changing anything at the moment. Then we're going to work a bit with the distance here, so we need something based on the location of our hand grenade. Vector math distance, and we need on the position of the dimer itself. Oops. So that's just a position. Alright, so now this output is the distance between the center point and any of the dimers in the Microtubule. So we can now say, okay, we only want to move those that are, let's say, within this circle radius. That would be sort of a nice visual way of intuitive way for us to figure out which ones are going to move and which ones are not. So that's very simple. We'll just add a math. So this distance has to be smaller than basically this distance, the radius. It's very simple. We grab the scale object, threshold, and now we can. This is now a Boolean condition. So this basically says, is the distance between any object here and this empty, is it smaller than, well, this scale? And that happens to be set up exactly so that it's the matches the visual outline of this circle. So let's plug that into our selection, because it's a Boolean selector, and let's see it in action. So if we change the offset, let's say move it X to that, and now we should see less than threshold distance. All right, let's move that around. I am missing something. What is it? Distance value less than. Okay. I forgot to click the relative. Yeah. So we have to set the object info from original to relative, and now we can see, yeah. So anything within our sphere gets deflected, and as we scale up and down, the effect changes. So here we can just take a little bite out of the lattice, a little hole. But now we've just chunked these over here because we have our constant offset, and we want to do some deflection based on this distance again. So let's just do a kind of white noise offset. So we'll start with some random 3D noise. So it's white noise. We plug this into the vector math, add, because this goes from 0 to 1, and we want it to go from -0.5 to +0.5, let's say. So now it's a balanced noise. Let's scale it so we have a little bit of a value to play with how strong we want the effect to be, and then we need something to do something with this and this value. So let's chug in a divide, because we want the effect to be bigger the smaller the distance. So we need to somehow invert this value. So we divide by the distance, meaning smaller distance gives a bigger value for this vector. And then we simply plug this into our offset. So what do we have now? Let's make this a bit bigger, something like that. And now we see, well, most of them are affected, but the effect is a bit too small, so we can scale it up. And there we go. We have some kind of random deflection. We can make this even bigger, knock them out even further. And just like that, we can sort of make a hole along the microtubule underneath, for example here, showing a lot of, it's just hanging on by a thread, maybe a bit clearer even if we render it. Yeah, so then you can put multiple of these kind of objects in there, plug them in, and make your damaged lattice structure. Make a severed microtubule. It's a quick simple thing of just a couple of nodes. You can make something that's quite interactive. If you want to change the microtubule to something custom, you can easily do that. So let's do a quick little demo with just a fun example of what we can do. So let's delete our poor little cube, and we'll add a basic icosphere, and let's scale it up a little bit. Oh, quite a bit. So, and let's call this now centrosome. Centrosome isn't a solid orb, but let's take some artistic liberty here and make it work. So, what we want is we're going to add some splines onto this icosphere, and then we can add our microtubule model to it. So, let's first load a microtubule to get started. Take our asset library, load our microtubule, and there we go. Okay, just so we have a sense of scale. Yeah, that looks about right. And then we need some kind of node set up to get some splines onto this. So let's get started. Go to geometry nodes, home, to, new, we'll call this. Oops. There we go. All right. Add no, not that. We'll add a join geometry. There we go. We need instance of points. Check that in there. We'll need a polyline. Fine. Good. Curve primitives. Curve. Oh, it's called curve line. All right. Instance that. Minus 20. Yeah. We need to get a distribute points and faces, so we add all these at a given density. We'll turn this surface into a mesh. Add the rotation. Oops. Add the rotation. All right. So now we have some kind of crazy spiky looking thing with sort of randomly distributed points. Turn the density down because otherwise my laptop's going to explode. That's a bit too not quite enough. 1, yeah, that might be good. Maybe also make them a bit shorter. Minus 12. Minus 8, let's do that. I mean, it's not going to be a fantastic looking thing, but it's just to get started. Alright, let's move this around a bit. Now, just for a little bit of visual interest, we'll make these lines a little bit bent and a bit more random. So to do that, we'll realize these lines, so they're now separate things, and then we curve handle positions. Oh, no. I want to set curve handle. Curve set handle positions. Alright. We also need this right now is a polyline, so it doesn't have any curvature to it, but we can set curve spline type. There we go. Make it a Bezier curve. Alright, so now we have handles on it. We want to change only the handles for the endpoints, so we go endpoint selection. And there we go. So now if we change this offset, you can see we're changing it for both the handles of this control point and the one that's on the surface, but we only want to change this one, I reckon. Oh, it doesn't make much of a difference here because we only have these 2 points. But so we get something there, and that's fine, but we probably want to add a bit of randomness to it. So we go white, grab some white noise, vector math. So we have a little bit of a weight value. There we go. So now we can get some random curvature on it, but like this we see it's kind of seems to be going one way actually towards the positive coordinates. That's because this white noise is outputs between 0 and 1 and not negative values, so we need another vector math. Oops. When we subtract 0.5, and now it's more random, more balanced, as you can see. Good, so we've got our slightly crazy looking curves out there. And now we can simply actually drag in our Microtubule geometry nodes, this whole thing, as its own group into this scene. And if you just search for it, Microtubule, there we go. Right there. So we can grab the curve here and let's do some basic setup. I will take the okay, back to the asset library and drag this one in, the low polygon version, simply to save my computer some trouble because if I take the full resolution model, my computer might spontaneously combust. And we'll set that as the dimer model. And now I can connect it and we should see something. Oof. There we go. I see the problem is I didn't apply the scale. So, here's an important thing before we connect this, which will add a lot of geometry to the setup. I forgot to actually apply this scale on this object. So you click Control A, apply scale. So now this is actually scale 1. And we have now check our density again because it, as you can see, the density is now way too high. So we'll go set this back to something much lower. Let's try that. And we have to also apply this scale and then change our value again to get some randomness in there. Okay, I reckon that looks good. Now we can plug in this into the join geometry, and there we go. Oh, it's a bit laggy because my computer is having some trouble managing all of this, but yeah, it's looking alright. Oh, I see another small error. I reckon I should make this positive 66. Yeah, much better. So now we have our sort of centrosome looking thing. I mean, the ends look a bit strange. I would tweak these parameters, but as you can see, we have a sort of centrosome-like object with microtubules coming out of it. And you know, you could then tweak this, make this more to your liking, make this a little bit flatter because now it looks a bit too bent. 10, now they're a bit straighter. Or lower the density. Improve performance a bit. Anyway, that's just a little example of a very simple setup at combining the Microtubule model with some other geometry nodes stuff to make a funny looking shape. Yeah, look at that. There we go, we got a little palm tree island. Cool. When you download the assets, you'll also notice that there is an alternative Microtubule geometry node setup that is compatible with 3.0. This is actually the model I started out with, sort of building, but then I ran into some problems that I at the time was not able to overcome. Maybe if I were to spend a lot of time on it now I could, but this model is compatible with 3.0, so that means if you don't want to download version 3.1, which at the time of recording is still in beta, you could use this in the meantime. Compared to the 3.1 model, it has actually one advantage, namely that it can have, as you can see, two ends. It can have two. It can have a minus end and a plus end on the same microtubule. There is a thing for that in. Of course, you could in the new model you can flip the orientation and make it your plus end essentially minus end just with a toggle, and you would never in practice probably have these two ends that close together that you would be able to see them in a realistic scene, but still it's nice to have. The node setup itself is shown over here. Also quite a lot going on here, but most importantly, if I just disable the instance dimers, I'm in drawing mode again. Disable that and curved points. You can see the ends are curves, just individual curves, but the lattice are these helices, short helices set on the backbone on the backbone, and it's a different approach to what I've done in the 3.1 model. It's easier in some ways and more difficult in other ways. It generates new problems. The main problem with this model compared to the new one, and why you would use the new one over this one, is that here the dimers are not individual instances. So that means you can't do, deflecting individual dimers based on some other value, or put molecules only on some dimers, or even do the GTP hydrolysis, because the dimers are not individual instances. I think each helix is an independent instance, and all the dimers on it, but not the dimers itself. So that's the main problem of this, that somehow I'd either end up losing the ability to orient these dimers, so I couldn't get the orientation right, or I'd have to put a realize instances after putting the dimer model on, and that's a very bad idea, because then suddenly all this very complex mesh that you have for GTP suddenly becomes a real object, and your computer will really start to slow down unless maybe you have a massively powerful system. You'll really feel that difference. So if I were to realize instances on this object right now, Blender would slow down to maybe 1 frame per second or something like that. It would be very bad. So that's the limitation of this model. But if you just want to have a microtubule in the scene and don't want to download the beta, this will do. So it has its own set of parameters that are quite similar to the new model. I tried to at least do everything that this model did there as well. The parameters are not necessarily directly comparable. So if you have, if you like a raggedness of 40, it's going to be different in the 3.1 model. But in basic use, it's the same. It's just a curve that you move around. Oh, I see there's 2 overlapping shapes here, but that's all exactly the same. And yeah, the approach is fundamentally different, because this was based on helices, whereas the 3.1 model is a single spline for the tip and the lattice, which on paper is a little bit more elegant. But I suppose there's some interesting things here as well about how it's set up. Right, so you can use it as a backup, but I would strongly recommend that you use the 3.1 model unless you have a very good reason to stick to 3.0.